Hello, my name is Tara Saxic, and I will be presenting on Lyme pathology. So again, my name is Tara Saxic. I am a bachelor's prepared registered nurse, and I acquired that from Liberty University back in 2015. In 2010, I graduated with my associates in nursing from Harrisburg Area Community College. Currently, I am in the process of obtaining my master's in human biology, also from Liberty University. I obtained a diploma in herbalism from the Center of Excellence in the United Kingdom. And work history, I worked as a charge nurse for inpatient and outpatient facilities, health coach, nurse case manager for workers' compensation, CPR instructor, and also trained home health aides. So an introduction to Lyme disease, we can read this together to begin. A little more than 30 years, Lyme disease, which is caused by the spirochaete Borrelia burgdorferi, has risen from being unknown to becoming a global public health problem and a prototype of an emerging infection. Lyme disease is transmitted through the bite of infected ticks whose saliva is a complex mixture of peptidic and non-peptidic molecules that aid in engorgement. Tick-borne pathogens are transmitted in tick saliva during blood feeding. However, saliva is not simply a medium enabling pathogen transfer. Instead, tick-borne pathogens exploit saliva-induced modulation of host responses to promote their transmission and infection, which is called saliva-assisted transmission, or SAT. The bacterium is highly reliant on the host to survive, as the bacterium lacks many central metabolic pathways to generate its own nutrients. This disease process through 3 stages: early localized, early disseminated, and also late stage disease, also known as chronic Lyme. To better understand the pathology of Lyme disease, especially in its ability to establish persistence in vertebrate species, including humans, the progression of the Borrelia burgdorferi, the Lyme bacteria, from its reservoir in the tick to transmission into vertebrates host, and to its localization and persistence in neural and other tissues are key steps towards eventually finding means to intervene and resolve the infection. So moving along to the pathogenesis of Lyme disease, early studies focused on identifying the causative agent. So as they found the Lyme bacteria and began to understand the disease is transmitted through tick bites, the diagnostic challenges were limited at that time, and also diagnosis and treatment were delayed, with some going years before diagnosis. Acute, subacute, and persistent Lyme disease is an inflammatory disorder composed of pathologically of lymphocytes, plasma cells, macrophages, and mast cells that have currently been discovered with Borrelia burgdorferi. The tick-borne Lyme disease pathogen also produces glycan chains and is occasionally, should I say, replaced with an unknown disaccharide. The lymphoplasmocellular infiltrates can sometimes be seen in the skin, subcutaneous tissues, but also in the lymph nodes, spleen, liver, myocardium, brain, automatic ganglia, and peripheral nerves, which later on we will touch on how vast the symptoms can be and why this is hard to diagnose. So the joints in arthritic cases have proliferated synovitis, fibrinaceous deposits, the lymphoplasmocellular aggregates, and also mast cells. Varying degrees of vascular damage occur in these sites. However, usually only in late or the chronic stage of the disease. Now, serological assays and immunoblots are now used as the primary discovery method. Further research has uncovered that the Lyme bacteria can sense whether it is located in a tick or mammal and adapt its responses to environmental signals such as temperature, pH, oxygen levels, carbon dioxide, but also nutrient availability and reactive oxygen species. The bacterial replication rate affects expression levels of numerous infections associated genes and also proteins. The carbon dioxide is important in determining the virulence of the Lyme bacteria in mice. And also the Borrelia oxidative stress regulator plays a pivotal role in establishing mammalian infection. So the Lyme bacteria can grow and survive without iron. Genes generate an oxidative stress response that is involved in the transfer of manganese, excuse me, and other metals. And then the use of bioluminescent Borrelia as a tool for studies in mice allows the visualization of the kinetics of infection and also with different strains of the pathogen, and enables real-time evaluation of gene expression in the skin, the heart, also the joints of mammals infected with this bacteria. Now, futuristically speaking, emerging themes regarding the pathogenesis of Lyme disease include one being the Lyme disease encodes several multifunction proteins. Two would be that it also encodes several distinct proteins that may inhibit host complement activity at different stages of the cascade. Also, three being the immune response to the Lyme bacteria are not only responsible for controlling bacterial numbers, but also the disease manifestations. And then, of course, four, it encodes multiple adhesins that play distinct roles in the infection process. Onward to immunopathogenesis in Lyme disease. This contributes with T cells to host defense during the Lyme bacteria infection, and it remains incompletely defined, to be honest. Early studies suggested that the induction of the Th1 responses could drive the transient inflammatory pathology seen in mice that are infected with the bacteria, and that also the dominance of Th2 responses attenuated that response. Researchers also isolated dendritic cells to understand what contributes to a weakened immune system in post-Lyme infection. So the dendritic cells were found from healthy study participants who were exposed to the Lyme bacteria. And what they found in these healthy participants is that the bacterial infection causes receptor sites on the surface of these dendritic cells to mature and become active. So these cells normally present antigens to T killer cells, the immune system agents that remove the invaders from the body. However, the researchers found and believe that when the dendritic cells interact with the Lyme bacteria, that they're structurally changed and keep the dendritic cells from marking the Lyme bacteria proteins as foreign. So as a result to this, the dendritic cells attack the T cells, but in response to healthy cells in an area rather than against the microbes of Lyme disease. So because of this, the researchers further surmised that if a person has a genetic predisposition to autoimmune disease such as lupus or rheumatoid arthritis, that the infection by the Borrelia burgdorferi may trigger their development. So they might be predisposed to those autoimmune problems. However, when the Lyme bacteria is triggered, it can actually turn on that immune response to cause an autoimmune issue. Humans appear to harbor low-level persistent Lyme bacteria infection as well. So the persistence seems to be a function of the bacteria's active immune suppression by forming antibiotic-resistant biofilms, which serve as a camouflage from the immune system and use evasion tactics that potentially lead to lingering symptoms. Now, the biochemistry of Lyme disease begins again with the spirochaete bacteria, and that's identified due to its corkscrew presentation. The unique genome of the Lyme bacteria has a linear chromosome and numerous plasmids, some of which encode essential proteins for virulence and also viability. The Borrelia burgdorferi has a very small genome compared to other bacteria. It does not encode toxins or lipopolysaccharides, but it does encode many lipoproteins relative to other bacteria. So it is also missing its genome, also missing from its genome, should I say, are genes that enable bacteria to synthesize such essential products as amino acids, but also fatty acids, enzyme cofactors, which are super important, and then also nucleotides. The Lyme bacteria depends on its environment to provide these nutrients and has evolved specialized mechanisms for adapting to different environments. So all these things really play a significant role in the detection, but also the treatment of Lyme disease and why it's such a challenge. Because these surface proteins undergo constant antigenic variation, which allows them to evade the host immune system. The bacteria can shift its metabolic or its metabolism towards glycolysis, and then this metabolic adaptation leads to lactic production, or should I say lactate production, sorry. And this will help it survive in the host. A crucial part of the innate immune system is the complement system, and this bacteria evolves its outer surface proteins to inhibit the complement system. It changes its cellular metabolism, and it has proven to be a central factor in driving cell behavior. It has been shown that the cellular metabolism of immune cells change when exposed or infected by several pathogens. So while this is often an adaptation of the host cells to the infection, sometimes it represents a mechanism through which the pathogens evade immune activation. The oxidative stress is produced by excessive production of mitochondrial superoxide and is believed to be the main contributor to damage within the affected cells, which can damage DNA, the proteins, and also lipids. So therefore, the bacterium likely interacts with host cells to obtain these metabolites, thereby affecting the host's metabolism on many different levels. Epidemiology of Lyme disease. It's really an endemic in several areas of the United States and Europe. The infection tends to occur during the late spring, summer, and early fall, which coincides with the nymph feeding, which are so-called baby ticks, I guess you could say. It interacts with their activity, and more people interacting in spaces that increases the risk of tick bites. So hunters being out in the woods, hikers, different things like that, but also people that are just playing around in their backyard. So it's quite prevalent, and in this area, we live in Pennsylvania, and it is definitely quite prevalent here in Pennsylvania. The life cycle and prevalence of deer ticks are strongly influenced by temperature, where they primarily are active when temperatures are above 45 degrees. So with that, if we had a mild winter, for example, tick activity can still occur. And they also thrive in areas with at least 85% humidity. So the risk of human infection is determined by the distribution of abundance of vector ticks, but also ecologic factors influencing tick infection rates, and also the human behaviors that I mentioned that promote tick bite. Infection rates are highest among children ages 5 to 15, but also adults older than 50. And in the northern United States, where the disease is most common, the exposure occurs primarily in areas immediately around the home. So while the exact number of Lyme disease cases are still being compiled for 2024, the CDC had estimated that around 476,000 people are diagnosed and treated with Lyme disease annually in the United States. So knowledge, of course, of this disease when it comes to epidemiology is important for patient management and also proper early diagnosis. Here we're looking at the signs and symptoms of Lyme disease, which earlier I had said can be very vast because of how many different systems the Lyme bacteria can invade. And so because of that, Lyme disease is often called the great imitator or the great mimicker, because like I said, those symptoms can mimic those of many different illnesses, even the simple flu, for example. And because of that, it makes diagnosis challenging. This is due to the wide range of symptoms and the ability of the bacteria to affect various organs and systems. Now, the Lyme disease is a bacterial illness, and it can cause simple things like fever, fatigue, joint pain, a skin rash, and more serious and complications such as joint and then also nervous system involvement. Now, Lyme disease is the most common vector-borne disease, as we have earlier surmised, and that is uh, kind of throws into the same category as diseases transmitted by mosquitoes, ticks, or fleas in the United States. But Lyme disease is transmitted through the bite of certain species of infected ticks that carry this specific bacteria that causes Lyme disease. The ticks live not only on deer, but also on rodents and birds and other host animals. Now, deer don't harbor the bacteria that causes Lyme disease, but specific other hosts, such as the white-footed mouse, does. And ticks pick up the bacteria by feeding on those infected hosts and then going and feeding on other infected hosts after the fact. So Lyme disease symptoms can appear within 3 to 30 days after a bite. However, research has shown that the bacteria is actually within the system within the first 24 hours after bite. Here on the slide, you can see the stages, stage 1 being early localized infection, stage 2 being early disseminating infection, and stage 3 being late disseminating infection. And in stage 1, it pretty much is surrounding the disease, begins with the bite of the infected tick, where the bacteria is introduced into the skin. And that's where we will see the well-known bullseye rash, but that's not 100% when it comes to diagnosing Lyme. There are several people who have no idea they have even been bitten by a tick, let alone seen a rash, to be able to tell the physician, that they had been bitten by a tick. So the rash can be common, but it's not 100% all the time that you're going to see that on a patient who has been infected with the Lyme bacteria. So with the rash, though, if you do happen to see it, because the bacteria will spread locally in the skin tissues first, and it will cause the rash to expand. So in the stage 2, though, if it's left untreated, the bacteria can spread through the bloodstream to other parts of the body, including the joints, heart, and also the nervous system. And these can range in a variety of symptoms as well, including flu-like symptoms, and that can include a headache or even a stiff neck, fevers, chills, arthralgias, muscle aches, and then also profound malaise and fatigue. When it comes to neural involvement, we can look and see meningitis or subtle encephalitis, cranial neuritis, neuropathy, ataxia, or even myelitis. And so Lyme disease doesn't only affect the central nervous system, but can also affect the periphery too. Cardiac complications are rare, but they can occur. And what we see in Lyme disease is AV blocks in various different degrees, but also myopericarditis and then also cardiomegaly. Now, in a late disseminating infection, which is stage 3, chronic arthritis, we will see, and so a lot of joint inflammation, typically in larger joints, which we can see there in the picture of the knee, and then also other neurological problems in stage 3, such as nerve damage in the arms and legs, because like I said, it can also affect the peripheral, not just the central nervous system, and also decrease in concentration, memory problems, sleep disorders, and all of these things could develop. So again, just proving how difficult it is to diagnose and then treat such an invasive, virulent infection. Diagnostic testing for Lyme disease it begins with the current CDC interpretive criteria for serodiagnosis of Lyme disease, involving a 2-tiered approach. And these consist of a first-tier enzyme immunoassay, which is also known as an EIA, or an indirect fluorescent antibody, which is called an IFA assay. And these are followed by the confirmation of positive or equivocal results by a second-tier immunoblot, like the Western blot, for example, which is very well known. Usually, the ELISA is the first tier that's used, and the Western blot is the second tier that's used for confirmation. But if we go to the next slide, we'll see the differences between the first tier diagnostic test, the EIA and the IFA. With the EIA, it relies on enzymes to convert a substrate into a detectable product, usually a colored compound, and this indicates the presence of the concentration of the target molecule. And like I said, the example would be the ELISA, which is, or the ELISA, however you want to pronounce it. And it's a common type of EIA to detect antibodies and antigens in a sample. So the advantages of that would be relatively simple tests, and the results are available pretty quickly within 24 hours. Now, the big disadvantage, and it is definitely a big disadvantage, is that the EIA is reported to only have moderate to variable sensitivity between 60 to 95%, which leaves a lot of room for error and also false positives or false negatives. And then you have the IFA. It uses fluorescently labeled antibodies to detect and visualize specific models or molecules, should I say, like the antigens in a sample. Detection is when the labeled antibody binds to the target antigen. The sample is illuminated and the fluorescence is detected. So advantages with that is that it's actually high diagnostic sensitivity and specificity, but disadvantages is that it requires specialized equipment and also expertise, which just raises the cost. So the ELISA, like I had mentioned, is the first tier well-known diagnostic test for Lyme, and it also detects the IgM and IgG antibodies against Lyme bacteria. So at the same time, if we go to the next slide, we'll see the differences between the IGeneX and the Western blot, which are both immunoassays. And so with the IGeneX, it uses a recombinant protein sprayed onto a membrane strip, allowing for detection of multiple species of strains of the Lyme-causing bacteria. And then it's also very specific, more specific than the Western blot, because it uses those recombinant proteins instead of proteins from a natural source. So the advantages to that is that the IGeneX test tests for multiple different species and strains of the Lyme-causing bacteria, and it's more sensitive, meaning more specific than even the ELISA, the IFA, and then also the Western blot tests for Lyme. It also only uses one test strip, and so that one test strip can detect the multiple species and strains. Moving to the Western blot, we'll notice that separates proteins by size and by using electrophoresis, and then transfers them to a membrane where antibodies are detected. The Western blot can be less specific than the IGeneX immunoblot because it uses whole cell lysates, leading to non-specific protein bands that confirm the presence and the presence of specific antibodies against the Borrelia burgdorferi. Now, these target proteins, when you look at a test, you're going to see small p and a set of numbers behind it, and those are to detect the target proteins, or otherwise known as bands. And it could be P18, P23, P30, 39, 41, 45, 58, 66, 93. But it also detects OSPA, OSPC, OSPF, and then also C6 antigens. Now, the only other tests that are sometimes used for this would be, and they're also less likely used, is other two-tiered testing, or second-tiered testing, sorry, such as the immune PCR and also metabolic profiling. Treatment for Lyme disease, general principles would be antibiotics. They are typically the primary treatment for Lyme disease, no matter if it's, a physician on the mainstream medicine side or a physician on the natural side. Pretty much everybody agrees that people who have Lyme disease need the antibiotic treatment. And most patients will recover after antibiotic treatment. However, there are definitely some that still have those lingering symptoms, and it's due to the fact of the ongoing inflammation, the triggering of autoimmune issues, and things that we had discussed earlier about how Lyme disease camouflage itself from the immune system, so it tends to linger. The recovery rate may also vary depending on the specific symptoms, and the recovery may take weeks to months after antibiotic treatment. When I had Lyme, it was significant. One day I just couldn't get off my bedroom floor. My husband had to carry me around, wheel me around in a wheelchair. I had trouble walking, talking, even at the point where I couldn't hold my head up. And that was also after I had already been diagnosed and on doxycycline for a week. So they admitted me. I ended up on Rocephin, came home with a PICC line, and the rest is history. And what they told me is that it could take out to 6 months to start feeling better. So I did a lot of research myself, and between mainstream medicine and natural medicine, I feel the combination of the two really helped me get me back to my baseline, and taught me a whole lot of extra things that can be done when treating different disease processes. So in about 10 to 15% of individuals with early Lyme, substances released by the dying bacteria can cause a brief worsening of symptoms called a Herxheimer reaction. And this can occur within 24 hours after starting the antibiotics. So yes, I had this, and I had no idea at this point in time, never knew the term Herxheimer reaction until Lyme disease. And when I was in the hospital and I looked at the doctor and I could barely even hold my head up because it hurt so bad, Toradol barely took the edge off. It was pretty awful. I also had a lumbar puncture, so I thought maybe I had a leak that was causing the headache, and it turned out that it was the Herxheimer reaction. So it was pretty significant. It made me photosensitive and everything. So, and like I said, it can start within 24 hours after starting antibiotics, and it continues for more than a day, but it does eventually resolve. But because of this, antibiotic therapy should never be stopped, even if the reaction develops. But it should continue. It should continue as initially planned. So no delay in treatment and no change in treatment at that point. There's no evidence that Lyme bacteria are resistant to antibiotics commonly used to treat the infection. In rare cases, additional therapy beyond the recommended doses may be used for a brief period, like typically a month. However, there's no evidence that further treatment with any antibiotics improves slowly resolving symptoms. Those with post-Lyme treatment disorder, which is more of an accepted term, and chronic Lyme is not, but it's pretty much the same thing. Now, this is typically not given additional antibiotics because the symptoms usually resolve within 6 months post-treatment. So how about that? Numerous herbal cocktails and remedies are also available for Lyme, and multiple physicians and naturopathic doctors have their own special protocols between the herbal cocktails and remedies and also antibiotics. So the goal is to eradicate the bacteria, obviously, which would be the antibiotics, while driving down inflammation, which could consist of changing your diet to, they call it an AIP, which is an autoimmune protocol, and that takes out all inflammatory food triggers for the most part. You stay on that for a little while, and then you start introducing foods back in. It's probably more predominantly known as the elimination diet, but that helps. And then also just different supplements that will help drive down that inflammation and bring it to a close. So these can include different herb preparations, like I had mentioned, the dietary modifications, and then also the supplements to support the body's immune system and also the detoxification pathways, and then, of course, lessening the inflammation. Now, as we see here, medication in early Lyme disease is a standard treatment for Lyme, and it's typically a short course of oral antibiotics like doxycycline or amoxicillin, and then I also have Ceftin listed there. And then with early disseminating Lyme disease, where it spreads to other parts of the body, then the treatment is similar to early Lyme with doxy and amoxicillin or Ceftin, and it's usually about 14 to 21 days, just depending. For doxycycline, it can be 10 to 21 days, going out to a month if needed. When it comes to Lyme arthritis, again, usually the oral antibiotics are out for about 28 days, but it's still the oral medication, no changes there. And then with neurologic Lyme, typically this is where we will find meningitis or peripheral nerve attachment, and also nerve block, and different nervous system conditions like that. And this is where the Rocephin will come into play, and the PICC line is inserted, and the treatment is typically 4 to 6 weeks. So the doxycycline is a tetracycline antibiotic, and it's usually the first line of treatment with Lyme disease. Didn't work very well for me, but we know why now. I needed the PICC line. And then the amoxicillin is a penicillin-like antibiotic, and it's another option for treating Lyme disease, especially in children, because it does come in liquid form, but it's also a pretty mild antibiotic. Now, Ceftin is a second-generation cephalosporin that can also be used, and as I mentioned, the ceftriaxone is a intravenous antibiotic delivered by PICC line, done in the home, and this is for more severe cases, especially those with neurologic involvement. So switching, because it's Lyme, you have to talk about the herbal approaches too. You just have to. The current finding is that the virulence and persistence of Lyme bacteria should be treated with antibiotic medication, such as the ones that I had already listed. However, a new study also showed that 2 antibacterial herbs, such as the ones listed here, the Ghanaian and then quinine, and also Japanese knotweed, show promise in activity against the Lyme bacteria. And this is because the active ingredient in the Ghanaian quinine is an alkaloid called cryptoleptin, which people have traditionally used against malaria and hepatitis, septicemia, and also tuberculosis. Japanese knotweed features an antioxidant called resveratrol, and some studies have suggested that resveratrol may have anti-cancer properties and may protect the heart and brain health as well. So in preclinical test tube studies, and this is as far as the study went, scientists observed that these extracts from the 2 plants prevented free-swimming bacteria from proliferating, even when they were present at low concentrations of 0.03 and 0.5. The 2 plant extracts also killed entire microcolonies of the bacterium that causes Lyme disease too. So the scientists noted that one 7-day treatment with 1% of the Ghanaian quinine extract eradicated the bacterium in lab dishes. Moreover, the bacteria could not make a comeback following this treatment. So it's definitely something to look into. At the same time, some of the other natural compounds with anecdotal evidence of effectiveness towards Lyme, such as monolaurin, which comes from coconuts, and then also colloidal silver, offered insignificant to no results using the same methodology. So while the current findings, they indicate that plants hold a promise in treating Lyme, the researchers caution that the studies in animals and humans are still necessary to confirm natural remedies' effectiveness and safety. And so that's why at this point, whether it's mainstream medicine physicians or natural medicine physicians, they both definitely across the board suggest antibiotic treatment versus just herbal remedies when it comes to Lyme disease. It's just too tricky and too virulent that you just don't want to miss any or let the symptoms go on longer than what needs to be. So here I put in James 1, 2 to 4 out of the New King James Version, and it says, "My brethren, count it all joy when you fall into various trials, knowing that the testing of your faith produces patience. But let patience have its perfect work, that you may be perfect and complete, lacking nothing." And so I think this is a perfect verse to end this with because, as I said, Lyme is virulent, and it can wreak havoc for quite a long time, even after treatment for people. And back on one of the slides, I had a little cartoon there of a guy sitting in the corner looking all beat up, and it said that he was in round 8,000, and that's pretty much what it feels like. Sometimes you just have a hard time seeing the light at the end of the tunnel, but then once you start feeling better, where you're having more good days than bad, and then all of a sudden you start feeling better for the most part the whole way around, then you realize that, again, just like this verse says, the testing of your faith produces patience. So thank you so much for listening, and I hope you have a great rest of your week.